what an illustrious panel I have with me with so much of experience around. And thank you, everyone, Kelia, Dr. Ranjana, Vipul Bhai, and Praveen for joining this so very important topic. And I'm sure we would have discussed this, but every time there's something new to talk about. And even when we were discussing and hearing about some of the earlier presentations, some of the questions were again on contaminations. And so people are really concerned about contaminations, whatever technologies we bring in, and how are we going to control. So, it's a long drawn process to discuss about it. But let me just ask all the panel members over here in short. How important is it to minimize cross contamination in the pharma industry? I mean, I'll take a quick round of these points for each one of you. So, Kilia, you may start this and maybe take a round with Kilia, Dr. Anjana, Vipul Bhai, and Praveen.
Sure. That we do our best to minimize any sort of contamination through equipment cleaning, through cleaning procedures, cleaning validation, by training our employees and all the things that we're going to talk about here in the next hour. But I would say it's very critical. It's something that the FDA looks very closely at. It's something that is like a hotbed point right now. So it's something that we all need to make sure we are focusing on.
Okay, Dr. Ranjana.
Okay, well, first of all, thank you for bringing this topic up again. And as you said, it happens again and again. And each time we talk about it, something new and something old. I just want to remind ourselves that this is going on since 1963, when it first came out in the regulation. And then it came back in 1978. And it's 2024, and we're still talking about cleaning validation, what's new, what's old. And as Kelia said, it's like super important because of the obvious reason is to prevent contamination, is to protect our product from ensuring it is safe and effective. So I'm looking forward to this discussion, Dr. Rajiv. Thank you.
Thank you so much, Dr. Anjana. Vipul Bhai, I know you are right now correctly listening, but thank you for joining this call.
Thank you for joining this call. Thank you. It's my privilege to have the panel with the media and so I think
your voice is breaking. I'm not sure if it's me or everyone.
Is clear now?
Am I getting a bit of break in voice?
Okay. Right. Is it okay?
Okay. Let's hear it.
I said it's my privilege to be a part of this with Kelia and Jana and so what I mentioned about this about the importance of the minimum and cross contamination. The one thing which I want to also that it's absolutely critical for the patient safety in product quality. Like is cross contamination can cause the adverse reaction, failures, any condition, entirely impacting patient quality. And start in the 1960s somewhere, but now over the time we will discuss during this time they have paradigm shift from the
request to use as audio is not low.
Okay. So try to do that. So now you can continue with the.
Yeah. So maybe I'll pitch in, and till the time Vipul joins back, I'll share my thoughts. So am I audible? Is it?
Yeah.
Okay,
Praveen, you are audible. Yeah,
sure.
please go ahead.
So it is extremely important in terms of minimizing the cross contamination for pharmaceutical industry. considering the fact that it has got a huge impact, and we're talking about two sets of impact over here. One is on patient, right? A risk to patient is huge. Also, the impact depends on the type and extent of contamination, of course, as well as the specific patient population which is being exposed. If I had to summarize the kind of risk coming out of contamination and why it is so important, is the first is very critical risk that microbial contamination, resulting into infections, toxic shocks, endotoxin related reactions, particulate contamination, which industry is always facing in terms of mechanical injury to internal organs, respiratory related issues. So we get all-time adverse drug reactions, allergic reactions, drug interactions, which are also very critical from patient safety point of view. Chemical impurities, degradation product resulting into toxicity, mutagenic effects, reproductive harms are significant. In a nutshell, it has got a short-term and long-term health consequences for patients, resulting into chronic conditions, uh, misinterpretation of symptoms, and several other things. So, from patient point of view, it is critical. But the second part of it, that from organization point of view, also is critical because it takes a lot of time and investment in terms of investigating the reasons for it. If the batch is out of the plant, a huge cost, huge investment, huge effort in terms of recalling it. And the critical part of it is that the medicine which we are looking for patient to have will not be available for some period of time. So it impacts on the drug shortage, it impacts on medically necessary products, and disrupt the supply chain. So from my point of view, it is very critical to minimize the cross contamination.
Thank you. Thanks, Praveen. I'm sure that this is echoed by almost all of you on the criticality of controlling the contaminations, the pharmaceutical.
So, Dr. Rajiv, am I audible now?
Perfect. Yeah, Vipul bhai, this is much better. Yeah, please go ahead.
Thank you. Thank you. Sorry, I just joined with my mobile, so something that went wrong in the hotel Wi-Fi. And anyway, so I was echo the same thing what everybody mentioned, that is cross contamination many times give the adverse reactions and treatment failures and life-threatening conditions. And then now what we are going to discuss about this cross contamination may occur accidentally transfer of substance, microorganisms, and other unwanted materials. And then, as Kelly mentioned, that FDA is more and more focus onto this, and then there are several warning letter, important letters are being given to the industries. So we need to really, as a company, to work with the regulator to overcome this cross contamination issue. That's my point of view.
Thank you so much. The next question I would ask to Kelia, Ranjana, and Praveen. See, regulatory agencies have been regularly supporting the understanding of contamination controls by way of publications, guidelines, and technical documents. What are some of the recent documents and practices or updates that these guidelines have come up with, and specifically on the products like sterile and non-sterile manufacturing? Kelia, you're on mute. Kelia, you're on mute.
Thank you. Now you can hear me. Am I audible?
Yes.
Okay,
Yes.
I'm sorry. Thank you for that question. I am going to point out quite a few things that if you want to take notes, like now might be the time to pull out your pen. So the FDA has provided several recent updates and has reinforced best practices for both sterile and non-sterile manufacturing in all of our guidance documents. So to try to streamline it for you, the key themes and best practices that are highlighted in the recent guidances are contamination control, contamination prevention, to make sure you use a risk-based approach. One thing that we'll talk about some more is the use of AI, automation, and some advanced tech, cleaning, validation, and verification, continuous manufacturing, which we've talked about today as well. And so just to give you a little bit more depth about that, the sterile guidance, which came out in 2004, I know this is a bit old, but these are things that are being reinforced. So I'll start with sterile and go down to non-sterile. So it's guidance called Sterile Drug Products Produced by Aseptic Processing. This was published in 2004. It talks about the best practices being the use of aseptic techniques and controls, which include personnel training, gowning procedures, environmental monitoring, facility design, but the use of advanced tech for example, RABs. So and even going a step beyond RABs, there's something that has been in talks a lot that we may or may not have talked about as of late in IPA are RMMs. RMMs are rapid microbiological methods. And these can be used in sterile manufacturing, which can be used to detect and identify and quantify microbiological contamination very quickly as compared to culture-based tests, which can take days or weeks, right? So this can enable you to have contamination control and improved QA and faster batch release. Of course, there's environmental monitoring and ongoing verification. One of the recent updates to this guidance is the use of robotics, such as like your clean in place programs and things like that. I think it is important to also remember that we can't just establish the CIP and say, this is it, and here's what we're going to do, and it's clean, it's verified, it's validated. You could also use things such as AI to go through the data that like you get from like your CIP, see if there's any holes, any inefficiencies, or anything that you can do to improve. So this is one way that we can use tech. Just one more guidance about the RMMs. It came out in 2019. This is the FDA's guidance. It's called Quality Considerations for Continuous Manufacturing. The focus of like this guidance is a bit more broad, but it does talk about continuous manufacturing for both sterile and non-sterile drugs. The best practices for manufacturing that it talks about are the continuous manufacturing processes, process and analytical tech, which is called PAT, which is used to control critical process parameters. And this employs a variety of tech, which includes spectroscopy, chromatography, process sensors, and chemometrics. This is also in line with FDA's quality by design, its initiative, which shows us how we can demonstrate a scientific understanding of our processes and how we can lead to more assurance that things are safe and cross-contamination isn't occurring. Just two examples for non-sterile manufacturing. Sorry, this is a little long, but I just wanted to make sure I gave you some points to go look at. So the FDA draft guidance for industry, which is called Quality Considerations in Non-Sterile Drug Manufacturing, also 2019. This talks about the best practices being cross-contamination prevention, cleaning, validation, and verification, and using a risk-based approach. And those three key things I just want to impart are so important, and it's what we're going to be using to apply across the board to all inspections when we evaluate cleaning. Those are going to be the three key things you always want to look at. Just to switch to the API, because I know there are some people here that are in the API manufacturing business. You can always look at ICHQ9 quality risk management, which came out in 2005. And really this just tells you to take a risk-based approach to your cleaning which is now very much required and something that we are told we need to look at during each inspection. Lastly, just one thing that is more new, came out in 2021, and this is for the control of nitrosamine in its impurities in human drugs. And the best practices from this stem from analytical testing and to using automated systems to track the impurity to track impurity levels during manufacturing. So these are just a few things that you can look at to see like where we are now. But if you want to know what we're going to look at once we come, I know that's the most important thing. What is the thinking right now on how we approach cleaning? You will find it here. This is the most up to date for us, and also the most up to date even beyond this panel. Look at the 43s that come out. Look at the warning letters that come out. This is what we're thinking, what CEDAR's thinking, and then how they tell us to go and conduct these inspections, right? And so a lot of times when like you look on the warning letters, you look at the 43s, they're concerning equipment cleaning and your environmental controls. Those are the two most important things. And with continuous manufacturing, you can cut down on like your risk because you have less interventions with people, with stopping the process, going from batch to batch. So those are just things that that are on the forefront of FDA, and hopefully you find that helpful.
Yep. I think that's what we discussed earlier on in the earlier two sessions. And I'm going to ask the same question about the cleaning procedures. So, but before that, let me hear from Dr. Ranjana about her impressions about what exactly are the supporting guidances that are helping the industry now. And specifically, I would ask that what are the learnings and the key focus areas of the industry on contamination control from the inspections, like what Kelia mentioned about learnings from the inspections. So has there been some learnings for the other companies as well? Dr. Ranjana?
So thank you so much for the question. Kelia has given us, I would say, the full gamut of all the guidances that are available, very clearly written. And honest to God, each time you read it, you get something different from it. It's so well written and it's so deep and thorough. So I encourage all of us to reread it. With that said, and Kelia hinted upon it a little bit, I think for industry, while the guidances are there, the most direct learning is from the citations, from the 483s and the warning letters. And what's new in the approach that the agency has taken, and this is my view and my view alone, but the information I'm going to be getting from is public domain available, is FDA didn't look at non-contact parts before. So it was only the equipment and what's in contact. Then we went really deeper into it. We started, let's say, like the fluid bed. We started looking at the inlet. We started looking at the outlet. Then we went all the way up to where it goes into the environment. We all have had inspections before, but we didn't have inspections where the agency looked at those parts. And I think, this is my view, the reason the agency is looking at it so deeply is because complex molecules are coming up. The same equipment is being used for a variety of equipment. Sometimes even, God forbid if people are using hormonal steroids and God knows what else, everything in the same equipment. So FDA is coming down really hard on us, the industry, and looking at areas which was not looked before. So I told you about the inlet, the outlet, I'm using one equipment. Second thing is the panels. So let's say your RMGs, your blenders, and the panels that are covering that, please open those. And you can see this in various 483s, warning letters. Open those panels, look at the gearboxes, because often what happens is industry has not cleaned that area from the time it has been installed. It could have been 20 years, it could have been 30 years. Another area which is completely new is the HVAC. Not just changing your filters, but looking to see if there is anything that is falling off of it. It sounds dramatic, but we've seen it, anything falling off and coming into the product. So, Dr. Rajiv, these are the new areas that are coming into purview and coming into inspection, which was not before. So it's the non-contact part that is really, really critical these days because we all know and concentrate on the contact.
Yeah. I think very well described both Kelia and Ranjana also that it's about the cleaning and how clean is something that we need to really look at. So Praveen, your thoughts about the current guidances that are helping us and what do you learn from the inspections that have gone with other industries and how are we getting alerted about them?
Sure. So I think Kelly has covered the guidance part of it. What I will summarize in terms of learnings for the industry. As Dr. Rajiv, Dr. Ranjana, you know that we are working on a subgroup on cross-contamination, avoiding cross-contamination, developing a best practice guide. So as a part of it, IPA has a database of last 10 years, and we have really, have dig down into that data. So what has come up is that around 334 observations are there direct on cross-contamination with recurring themes and more than 2,800 times the indirect linkage to the cross-contamination part of it, equipment, design, cleaning related, this thing has come up. So when we are looked into the whole data, there are 7 themes which are emerging, which are very recurring. I mean, and every year this is coming up in one or the other context. So these themes include cross-contamination linked to cleaning and maintaining the equipment, the procedures involved into it. How do you do the cleaning? How do you maintain the equipment? Gaps in handling and storage to prevent cross-contamination. That how you store the material, how you handle the material, and how it impacts on the overall contamination part of it. Failure in preventing cross-contamination of hazardous and potent drugs is one of the major theme coming up. Lack of adequate space. There are observations where space-related citations are there. Cross-contamination resulting into a chemical, physical change, deterioration, product-specific aspects of it. The majority of observation are into microbial contamination coming up because of the practices, because of the design and various aspects. And significant part of the citations are on inadequate procedures to prevent contamination. The kind of procedure design which is required across several stages is there. So key focus areas are definitely risk assessment, the design of the facility, the cleaning procedure, how do we train our operators in terms of ensuring the consistency of our cleaning operations, investigation procedures. While we do whatever, as an industry, we are moving forward in terms of environment monitoring, but still it remains a concern that how best we can have the environment monitoring program, indicating us that there is a minimum kind of a contamination. So the answer to all these learnings is that we should have a robust contamination control strategy, having this learning incorporated as a part of way forward.
Thanks, Praveen. That was quite useful for people to understand the importance. And the statistics that you gave is quite alarming in terms of the number, and you really look down at the numbers and the times that these observations were made. This is quite important that in the recent times, this is quite a common observation that is coming up. So my next question is to Dr. Ranjana and Mr. Vipul Bhai. See, in the recent times, as also Dr. Ranjana also mentioned, and Praveen also mentioned that we are having a lot of observations around the utilities. And there was a question I did purposely not answer the question or ask the question earlier on when we were talking about the earlier presentations. And this question was about the HVAC system. And so there have been a lot of discussions about how to control the contamination in those systems. And specifically now that we are moving into high potent products and where you have low dose products also. And even Kelia mentioned about the nitrosamines. So what has been the recent updates on the cleaning procedures that have been practiced by the industry and the individual companies? Maybe Dr. Ranjana and Vipul Bhai can take this question.
So I will take it first.
Yep.
Okay, thank you. So before going to this specific question, I want to just add about what Kelia and then the colleagues mentioned about this. The one thing we want to mention, like the guidances, and then Praveen and Ranjana, you mentioned the industry perspective. But one thing which we need to, my message to the industries and my personal experience, that how you convert your guidances into the practices. That is the main point. And that is where the industries are facing the issues. Like just now we have discussed about the HVAC cleaning procedures. Now is a significant paradigm shift in cross-contamination control strategies. Like we discussed about the QBD, but QBD is not only for the product, and it is for the processes, it is for the facility and design. So if you don't do the cross-contamination strategy from the starting from your the facility and then equipment and then come to the product. So I will give you example. The traditionally we were not having the mal and pal. Now, the newer design, especially for the OSPs and for other, there is a material lock and personal locks where the cascading, the air, the pressurizing the air into the systems. So, is cross contamination is only the powder or is other? So, according to me, is a man, material, and environment. These are the three main sources of the contaminations. So how we want to do in the quality by design. So first is the design of your facility where, as Prabhu mentioned about the what we call as the proper segregation. Like recently we were building the facility. Now the coming to the HVAC. So we used to have the what we call as a huge ducts coming out from the fluid bed dryer and going like it's almost 300, 400, sometimes 500 feet or it goes into the meters. So now the shift into the design is mentioned that there will be a continuous manufacturing, but I don't know whether everybody can afford that, but at least we can have the design in such a way that we can definitely going to reduce the ducts by which we can avoid the what we call as a contamination risk on this. Second point is that how much the design man and material we are going to go. Like a man moment, we used to have the primary garment and secondary garment, and we used to go, and then the same people used to come out and go, and there is a possibility of the carrying the contamination. So those are the things which is to be focused. The man mal and pal is must to do, where the mal pal can be the bubble or can be the pressurized or the what we call is a sink. So these are the based on the this one. Another point that regulator are agreeing, coming to the point of to the expense also, like they have agreed for the shared facility for the potent product, except some of the like cephalosporins or hormones or the cytotoxics. So the point of view of the potent product, as Ranjana mentioned, more and more products which are coming, which are the potent, more and more products which are coming into the more and more A&Ds are getting approved. Like you see that 42% of the product we supply to the US to that. So more and more A&Ds are getting approved. So, but coincidentally, all these products are more and more potent products. So, since the potent product come in the picture, the question is coming is how it will be cross contamination. So that is one point I mentioned about men. Then material movements, like how best you can design your material movement by the dirty equipment is segregated and clean equipment is segregated. The movement is segregated, and 99% you design the WIP or CIP so that the dirty equipment is not coming out. And then coming to the process where you have to control, like what we mentioned, that how eliminate the flying dust, how best you are doing your exhaust systems of the dust collectors. So these are the few things which we need to build in our design phase during we are designing the newer facility, or we have to upgrade the facility for that. So coming back to this HVAC, like we mentioned, the automation is a key factor. Like we need to see that, like prevention, automation, and real-time monitoring. As I mentioned, how we convert guidance to the FDL practices. I will give you one example. We have the troll ball is become a very popular into the duct cleaning. But has anybody has gone and see the how the troll ball is functioning? Has anybody gone and opened the ducts and see that what is going on inside? Has anybody seen that troll ball is chalked or not chalked? Whether a troll ball is there to do the 360-degree cleaning, whether this 360-degree cleaning is happening. So if you are not doing that, FDA is going to go and look for that, right? So that is what, whether whatever the water you are using, whether it is drain off, it is properly dried. So you are cleaning the powder. On the other side, you are contaminating the duct because of the improper drying. So I'm just giving one or two examples like that. As Ranjana mentioned, opening of the panels, cleaning of the this one is very difficult. The real practice is when you are opening the panel and you are see the powder inside is a bunch of all these wirings, bunch of all our automation like sensors. Now to do the cleaning of that, we need to really come out with some unique ideas how to do without disturbing. Suppose I do the cleaning of the panels, touching the wires, touching the sensors. I am going to go into the different issues like my sensors are disturbed. So whether I'm going to get the desired output because of those sensors. So these are the all the practical points which we need to really look into that when we are designing our cleaning process. So the risk assessment based cleaning process. I agree that we need to open the panel. We agree that we have got the citations. We agree that the citation is based on the powder or the accumulation of water can generate the microbial and contaminate this. We agree that, but how we can do that? That is a question which we really need to work together and ask for that. So more and more what we are discussing about this, more and more we are thinking that it is always better to have dedicated facility. But it is practical. No, it's not practical. So we need to really come out with the something which is very out of the box by which we satisfy the regulators, we satisfy ourselves that we are not creating anything which is safety and efficacy to the patient of this one. So like what I mentioned, like dedicated cleaning equipment, that is one. The QBD is another point. But more and more, I'm giving the more and more weightages on the how you implement your guidances, how you implement procedures, and second point is that how you monitor them. That is the most critical. That one is monitoring. Since you don't monitor, you don't know. If you don't know, you are not able to take the actions. Like we have the cleaning people. So we call as a cleaning crew. So we have dedicated cleaning crew. So they are dedicated for the particular piece of equipment. Like as you mentioned, continuous equipment or continuous complex equipment. Where to look for that? Where the powder will be? Because easy to clean is always there, but difficult to clean. How to do that? We need to focus on the automation of the cleaning. Like we use the pressure jet with a very high pressure jet, by which the powder which are there stick in somewhere that we are using. The other thing, the industries where they are doing the mistakes into the dry clean, they use the compressor. Please don't use compressor, never, because you are on the contrary contaminating other part. Always use the vacuum. Like regulators are always happy to see the vacuum, and vacuum cleaner is always available with the HEPA filter cleaners, or you can have the centralized vacuum systems by which you can avoid. So these are the few things which I want to make my point of view. And coming back to the potent product, I think that is where we need to really see that how the potent product we need to handle into the share facility. Like we have to do the FME based decision for the potent product. Like you have to dedicate the equipment with the isolators, and that is no other choice. That's my suggestion. But if you don't have that, you can, as I mentioned, the FMEA based checklist where you can drill down or open up your equipment, let's say compression machine, and then you identify which are the parts which are difficult to clean, and then how, whether you will dismantle and then clean and then assemble and then do the line clearance. So these are the few points which should be part of your FMEA checklist. And then the one point which I want that we are doing the mistake as industries, we are not dedicated the cleaning people. We have to dedicate the cleaning people so that they are doing more efficient cleaning if there is a human interventions are involved. And then in cleaning validation for the swab sampling, we have to have the prove the robustness by doing the 2 or 3 different cleaning people to prove that the human intervention manual cleaning also gives a desired output like what we are expecting. So coming back to the potent product, generally what we do that one is FMEA based cleaning, and then we also do the double cleaning. So type B is always done the 2 times rather than one. So it's a conservative approach, and every time, even though it is validated, we always do the swabbing from the different. So these are the few suggestions how to handle the potent product into the some of the area. Thank you, Rajiv.
Thank you so much, Vipul bhai. I think you gave so much of ideas of, I would say a very, very strong QA perspective about how the cleaning should be done and what are the pitfalls in the whole process. And you also mentioned about, the new technologies and what it can bring in terms of the compliance and the issues around that. So my next question would be to, again, to Kelia. I mean, we talk about all these new innovations in the equipment cleaning procedures. So have you seen any kind of a leverages in these technologies in the industry? And is the industry and the companies able to achieve the required and the desired cleaning levels with the use of these technologies?
Is this question is for me? I
Yes.
just wanted to
Yes.
double check.
Yeah.
Okay,
Yeah.
sorry, there's a little break. Okay. Okay, so for the question, I think that there are quite a bit of innovative technologies that can be used to make sure that you are in compliance with like the FDA standards and to ensure product quality. And I do think that some of these things are being done. I think there are ways that they could be improved. But here are a few things that I think based on the FDA's current viewpoint. Automation and robotics. So to automate cleaning processes such as a CIP system, which is a clean in place system, this can ensure consistent cleaning performance, minimize human error, reduce contamination risk, right? So these systems allow for a controlled, repeatable cleaning cycle with automated monitoring and validation. Robotic systems can be used in aseptic aseptic environments, such as an isolator or like clean rooms, which can reduce the need for human intervention. Advanced analytical tools, which I also briefly talked about the PAT, which is the process analytical technology. So this allows for the real-time monitoring of cleaning processes using PAT, which can track critical cleaning parameters like flow rate, temperature, chemical concentration, things like that. And these tools can also be used to analyze residual levels on equipment surfaces. So like we can ensure a thorough cleaning. One more way that we can use this is one thing I talked about before were RMMs, which are the rapid micro methods, rapid micro methods. Say that 5 times fast. These advanced tools can allow you to faster detect microbial contamination and can provide you more immediate feedback on the effectiveness of cleaning. Using AI and a machine learning. So AI can be used to analyze historical cleaning data to optimize cleaning cycles and to also predict your preventive maintenance program. So when do we need to clean our equipment and how often? And the predictive maintenance approach will also help to minimize unnecessary cleaning and to provide to make sure that cleaning is done prior to when contamination would occur. Single use text. So like for some processes, you may be able to use single use equipment such as tubing. This can reduce the need for extensive cleaning validation. If it's discarded per use, I know that can be a lot. And I also want to say this goes off, this goes back to what Mr. Doshi was saying, is that having a dedicated manufacturing base or line, those also help to cut back on cross-contamination. Having a continuous process, which we talked about at like the start of the day, that helps to decrease these things as well, right? There's so many things. So by integrating these innovative techs, you can ensure consistent, high quality cleaning performance, and you can reduce your contamination risk and also enhance your regulatory compliance. Thank you.
Thank you. I think, Kilia, you brought a very important aspect about predictive maintenance being also continued with the cleaning procedures, and also about the single-use accessories. I know that many companies do that because it's very difficult to have a dedicated facility, but accessories and things like tubings, pipings can always be dedicated, which can be changed when there is a product changeover. Yeah, that's a good point. Coming back to one more very important aspect about the cleaning. So I asked this question to Dr. Ranjana, is about cleaning validations and method validations. So how important is it to develop, validate, and the cleaning procedures, and also the testing methodology as part of the technology transfer? Because that's something area we normally do only for product transfer, method transfers, but cleaning procedures and cleaning methods is something quite important. So how do you think it's important for method transfers and technology transfer of these two very important aspects?
So I think, as we've heard a couple of minutes ago, different methodologies being used for cleaning. So if you're not using robots and you're not using automation, and most of us have not reached that stage, I think it's very critical to factor in time and effort in trying to establish cleaning validation and method validation during tech transfer, because the site is different, people are different. Even though the Uber management may be the same, the equipment, even though it's different, or it could be the same, but is the size the same? Is it exactly the same material of construction? So there are lots of variables. As it is, we know cleaning has so many variables. On top of that, if I'm going to transfer from site 1 to site 2, I better have my cleaning strategy in place, written with regards to how am I going to clean, what's the molecule that's coming in, understand the solubility of the molecule, understand everything that I need to know about the molecule. And is it going to be automated cleaning? Is it going to be a hybrid? I love the example of the troll balls. Am I going to go in and see, because those gaskets full of powder, so we need to go look at those things as well. And this is another opportunity for us, the industry, when we are going from site 1 to site 2, is even if you're doing the same thing, what you did in site 1, re-look at it, because things may have changed. And it gives us an opportunity to go back and revalidate, because the expectation from the FDA is to revalidate from time to time. What better time could it be when we're doing our site transfer? Method validation, I cannot emphasize how critical method validation is, because we're looking at low-level contaminants, if they're present or not. We're looking at unknowns. So method validation with the proper equipment and your LODs, your LOQs, have they been established? Your linearity, has that been established? Have you done proper macro calculations based on, again, your molecule, based on your PDE, All of these things must be looked at again during site transfer. Do not overlook it and don't think that it's okay not to do it because you've done it in site one. It ain't going to fly. So it's extremely important. Don't forget the linearity part of it, because sometimes that gets forgotten because you think you don't have to do the upper range because you're looking at low level, you're going to get dinged. So let's do a complete method validation. The method has to be specific. It has to have recovery. You have to show recovery in your method. Hopefully you're getting recovery 70% and more. And let's get stronger with regards to site transfer.
Absolutely, well said.
So, Dr. Raju, I
yeah.
just want to add,
Please.
if I may,
Please.
I allowed, yeah.
Please go
So,
ahead.
Ranjana, you raised a great point, and I want to just to help the industries. That the recovery is one point. Simultaneously, selection of the swab, that is
Yes.
the most critical part of
Yeah.
the method
Location.
validation.
Location.
That is where the we as the industries are facing a lot of issues,
Yes.
like non-leachable swabs. Like we have the unknown peaks, and then if you see that the once the FDA walks in, they go into the laboratory and then they see that ask for the unknown peaks or the this one. So the these unknown peaks may be because of the swab leaching. Now, if we don't use the proper swab, then we do the continuous investigation, investing over time, and so the selection of the swab, which is non-leachable swab, that is very very critical. Another point is that the swabbing area, then the recovery, as Ranjana mentioned, I suggest that the recovery should be all the MOCs of the contact parts. Irrespective of what, like in the fluid bed dryer, you have 7 different contact parts, different material of construction. You have to do the recovery from all the contact parts to see that. Yeah. Another thing which we are doing the mistakes that we take the swab and we put into the diluent, and that diluent may not be suitable for the solubility of that compound which we are targeting. So you have to select the diluent where your swab sample is soluble, or in this which you can record like more than 70%. So some of these points which we need to really where the industries are failing and we are getting the citation. And one point which Ranjana has mentioned that is very critical, and then Kelly also mentioned the new technology. So that also very critical. Like your potent product, your PD values are so low, your MECO is become so low, you have to go to the LC mass. So that is something which we need to do, go for that. And then one point is the expectation is that LOQ has to be 50% of your MECO value. So that is where the all these points. So Ranja, I want to just add what you are trying to mention.
That's very good. I forgot about the swab. It's extremely important.
Absolutely, absolutely.
And also don't let us not get fooled by if rinse is enough, because we have to do direct and indirect sampling. In some cases, rinse may be okay, but not most of the cases.
Not always.
It's
Yeah.
extremely critical.
Yeah.
Thank you, Vipul. I forgot that.
Yeah. So coming back.
No, I'm just trying to add to your
Ilya has,
point.
Ilya has a question. Ilya
Yeah,
wants to
please
add
go ahead.
something.
Yeah, Kilia, please go ahead. I saw you doing a thumbs up, and I want you to add to what.
Ranjana was hitting the perfect points. I was like, yes, she's
Yes,
gonna pass this her inspection. Yes, I was going like this. Great job. I do just want to add a few extra points before we move on. I know we don't have a lot of time, but I just want to give you from the FDA perspective, things like, here's kind of like 5 whys that you can kind of ask yourself and try to apply, because this is something we're going to be doing and trying to apply it once we come to like your site. All right. So one of them is, do you understand your process? You also just brought this up. Can you understand your process enough to make the SOPs, to make the steps, to know, do we need rinse and swabbing? I always say both whenever you can. But you understanding your process, knowing the ins and outs of it, will also allow you to be able to ensure cleaning process consistency, right?
Mhm.
So when you're transferring tech from it's between sites from like development to like to the commercial scale, it is vital to maintain consistent cleaning and and testing standards. And like we said, it starts with the design. It starts at the very beginning. It's not once we start to make it, now how do we clean it, right? Number two is preventing cross-contamination. So we want to ask ourselves, does like this set of cleaning these steps for cleaning actually clean, prevent cross-contamination, remove it to acceptable levels? We talked about that last time. What does clean mean? Have we defined that? It allows you to be in a compliance. This is a why too, right? So which means, so how can we be in compliance? That means we need to make sure that our cleaning processes, our cleaning SOPs, our cleaning validation does ensure compliance. And I mean, like, you can ask yourself, does this science-based or in a science-based way show that we can remove it once we clean it? And I do like the idea of having teams. I think that's a great idea, especially for hard to clean. I hope to see that more. Wink, wink. Taking a risk-based approach. Have we taken a risk-based approach? Do we know the score of like this particular drug, molecule, whichever we have, right? Have we done a risk assessment? And how do we clean this equipment? The hard to clean places. We do see a lot of firms where like they'll just have a list of places to clean, but where's the diagram? How do I know that this is the hard to clean space? Because I'm going to want you to show me, and I'm going to want you to show me like how you address that. And then last is efficiency and knowledge transfer, because what is determined and what is found in R&D should be clear and understood by our staff at the commercial site, right? So how do we train? So how do we do a thorough validation to make a smooth transition during our tech transfer to reduce the downtime, the need to rework, or product loss, right? So we need to have a shared understanding of our critical cleaning processes on like both ends, because they can help you to refine once you get to your site. And like she just stated previously, whenever you have a cleaning validation, you could have the exact same setup at site A, but at site B, you have to do it again. You have a totally new set of employees who may not understand the SOPs, et cetera, et cetera. So just to kind of round this out, those 5 things that I just mentioned are things that we're going to look at, things that we're going to see from like this perspective. We'll come into your site and ask, how are these criteria met? And if you have a hole in that, you're going to expect a 43, right? So these are things that you can take from this discussion, because all of what everyone said today was entirely correct, and use these ideas and you can prevent this at your site. Thank you.
Thank you so much. I think that was a great discussion, and thanks for chipping in with all those new insights also. In the recent times, I've seen that there are a lot of products which are being introduced, which are so difficult to clean. I don't know what happened. Suddenly, there's a spate of new products being introduced, where gone are the days that the equipment could be cleaned with simple water or conventional methanol acetone or ethyl acetate. But these are products which are just not able to get cleaned. And to make them the equipment to clean, there are another host of new cleaning agents being introduced by several companies. Now, this question is to Dr. Anjana and to Dr. Vipul also, and maybe I can also extend this question later on. Like, what is the care that needs to be taken when you introduce such cleaning agents in the regular operations? Because they themselves would pose some problem, or is there something that needs to be done? And maybe Vipul bhai, you can just touch upon the very basic question that comes up every time is about selection of a target molecule for cleaning validation, which is also very important. And people miss out the very important aspect about how to really select a product which actually encompasses all the products that are there. So let's hear about it. One is about the new cleaning agents and the cleaning procedures that maybe Dr. Anjana, you can start.
Okay, sure. So I think we'll start with the molecule hardest to clean. And I think whole host of molecules all of us have in our companies. And we need to evaluate and find out which is the hardest to clean and why is it hardest to clean? Is it the smallest batch size? Is it the largest batch size? Is it very complicated equipment? And I'm speaking in very simple terms because I know we have a large audience with using different kinds of equipment. So that needs to be evaluated each time you bring in a new product. Supposing we say molecule A is hardest to clean, and then you only have A, B, C. But if you bring D or you bring E, go back and look to see, is E harder? Or if it's a single entity, and then you have the same entity with another molecule, like it's a combo, don't think that that will suffice. You will have to go back and then prove that the molecule which has two in there needs to be re-cleaned and proven that this is hardest to clean. It sounds a little complicated because it is a little complicated. With regards to the detergents, now we have detergents, we have abrasive solvents, we have these reagents are sometimes harmful to the person using them as well. So I think that personnel also has to be protected from these solvents. And as you said, Dr. Rajiv, that there are many vendors. So I think it's a constant evaluation, is which molecule do I use, which is hardest to clean, and then how do I clean it? Is it detergent? Is it plain water? Is it the pressure? Is it the force? What is it, and how am I going to clean it? Or do I need to get some vendor? Because there are quite a few. I don't have names of them on top of my mind, but it's important to evaluate what's out there in the marketplace. And I would like to hear from Vipul. I don't want to hog up all the time because I'm conscious of the time. We have only 2 minutes left. So Vipul, why don't you come in now with regards to the various agents?
So Ranjana, great. I want to just build on what you mentioned. I want to add one thing on which for the selection of the cleaning agents. So as we mentioned that the harsh cleaning agent is killing your equipment. So first and foremost, which is required to perform material compatibility studies, which is not difficult. So material compatibility studies to prevent equipment degradation or corrosion over the time. So that is the first point which I look for the selection of the detergent. So, I know that many of the detergent in the industry which we are using is very highly acidic because it is that. But there are many cleaning agents now available which are the neutral in pH, but that is a surfactant. So, if you use the surfactant-based neutral cleaning agent, it is very easy to clean and it is not damaging your equipment. That is first point. Second point, we need to see that how easy to clean the cleaning agent. That is another point which we need to see. I've seen in industries when I'm doing my own audits that the detergent itself become the pain to clean itself because we have to first develop the method to detect the detergent because we have to have the macro level of detergent up to the non-toxic. That is one thing. And then we use huge amount of water to clean it. So if you use a surfactant-based neutral pH type of cleaning agent will be helpful, which is what we use. But I'm just trying to give you the industry. That is two things because we have to ensure during the cleaning validation, not only the your active substance, we have to also see the microbiological contamination, and we have to see the detergent or the any cleaning agent which is used. So these are the three things which we need to really see that by selecting the detergent. So that is one thing which I want to add because when we do the cleaning validation, cleaning validation is not only for API, also for the microbial contamination as well as the detergent contamination. Another point which we have discussed that when we do the detergent selection, the operator safety and handling requirement also is very critical. So that we need to see that it is not very highly alkaline or acidic. So that is something which we need to really on focus onto this. Now coming to the selection, as Dr. Rajiv you mentioned, now this is a very critical point, and Ranjana has touched best. So how many times we have discussed with our operator that where you find difficulty in cleaning, which product you are find difficult in cleaning. So operator interview is very critical, even though it is a highly soluble API. But the formulation is very difficult to clean because of, let's say, guar gum or because of some polymer which is used for the making the slow release products. So we need to really discuss with the operator, have their feedback, which are the difficult to clean. So my first focus is always the difficult to clean product. Then it comes to the solubility, then it comes to potency, and then it comes to the toxicity. So these are the four areas, selection criteria which we do. And then once we do these selections, the primary is the difficult to clean, means cleanability. Second is toxicity, potency, and solidity. Then we do the all the groupings, equipment grouping, product grouping, and so forth. But my target molecule is always based on these four. And then first and foremost, my requirement is always to discuss with the operator. And when we take the engineering batch, when we introduce the new molecule inside, when we take the engineering batch, during that time only we determine how difficult to clean this product is. And then whether I can continue with my validated cleaning method, or I need to revalidate the method because of this difficult to clean. So these are some of the points which I want to add to you, Dr. Rajiv.
Thank you so much, Goodbye. I think this was quite insightful and informative. My next question.
One thing is, sorry, just one point I forgot. We were discussing about the operator interventions. So we need to really say, like, automated cleaning processes, how many industries can afford and put. So operator intervention we're going to do, but we can definitely go with the HER formats. That is human error reduction formats. So it become so easy to operator to follow that format, what is to do, how to do, where to do. So, so because if you see the many citations are there that they will not follow the SOPs, they will not follow the checklist, something like that. So if you implement this HER format, and it is very useful to the operator to follow that, so that mistakes can be avoided. I want to just add one point. Thank you.
Thank you so much. I think we have been keeping Praveen quite silent for a long time, but he's been nodding. I know, in between both Kilia and Vipulbhai and Dr. Ranjan also mentioned about the cleaning procedures and the operators and the consistency of the physical activities that are done. So what would be your call? And especially I was more interested in your designation, which says Head Global Quality, Sustainability and Quality Culture. So I want to understand if you are really implementing those things in your as a designation and as your responsibility also. So talk about how are you making sure that your operators are really able to maintain the cleaning procedures, because you may have a lot of SOPs, you may have a lot of recipes, but how do you ensure that the people are consistent? And we discussed about having a dedicated team. So your thoughts, Praveen? And maybe
Sure.
Kilia, you can add a little bit on what exactly would you expect in your inspections on this aspect. Praveen?
Sure. No, I was really enjoying the discussion, and these are the inputs which are going to come into the best practice guide. So it was useful for me to take this discussion forward. Operators, I think, most critical part of the whole process. The more we work with them, right, it is going to be useful to ensure the consistency in terms of the cleaning procedures. So while every organization will have a training plan, what we are looking at is a very comprehensive training plan, which includes the overall package. It's not about the delivery of training. It is about having a comprehensive cleaning plan as a part of this package. with their understanding the layouts, equipment assembly, disassembly, tools, cleaning agents as per the cleaning validation process. A clear and very site-specific stepwise SOP, including pictures and videos in terms of how do we do it. Demonstration is going to be key. The more we demonstrate, I think it will be very useful. We will also have to support in terms of the correct and quality kind of equipment, such as non-pointed brush to avoid dent and scratches. This is very basic, but over a period of time, the way you use it, it's going to impact on the overall process. So, operator qualification, cleaning. While we do qualification for aseptic area operators and media fill and everything else, the strengthening of operator qualification program for cleaning operators is also going to be necessary. We will by touch based on one point that we will get more of the inputs from the operators. So I think speak up culture is going to be a key in terms of getting the inputs from site and where people are having challenges and how do we convert those challenges in terms of our opportunities. But I would just touch base based on the previous discussion in terms of opportunities in automation, because while there will be some manual work which will continue, there are new things which are coming up, and all of us are looking forward to it. Right, there are portable, intense pulse light disinfection systems where we get 6 log sporicidal reduction, no risk of chemical and residue. I think industry is working on that. Automated visual inspection systems with high resolution cameras and AI-based technology where we don't need to see visibly clean. I think system will determine visibility. So it has to be a balanced way. The more we focus on shop floor, more we handle operators, understand their needs. I think it will be more consistent result in terms of effectiveness of cleaning.
Thank you so much, Praveen. This was quite useful. Maybe between you and Dr. Ranjana, you can talk about, you mentioned about the new best practices document which is coming up on cross-contamination, and how is it shaping up? And do you think that this particular panel discussion has given you some more ideas about what to be included in that? And I'm sure that the discussions about the best practices on engineering and the PAT, this guidance which is coming up and getting published in the next couple of months' time is going to be really helpful for the industry people. And we look forward for your inputs. And Dr. Ranjana being a mentor on that group, and you being a very active member, we expect a lot. So just you and Dr. Ranjana, what is coming up?
Sure. I think this discussion is extremely useful, right? Because this is what the industry is looking forward to. As a part of IPA, I mean, I'm grateful for this opportunity. As a part of IPA team, what we are looking at to resolve the practical issues which shop floor is facing, right? So the approach is to have a lot of case studies coming up from regulatory agencies, case studies of individual organizations, knowing the equipment and instruments better. How can we reach out and know these equipments and instruments in a better way? Guidance for conducting risk-based assessments for identifying contamination-related sources, detection mechanisms. I have touched based on some of the automation-related aspects. So we are in touch with external consultant in terms of giving us inputs in terms of how the industry is moving, what kind of automation is coming up, and that will be also part of the recommendation. Contamination controls, specifically in terms of manufacturing controls, product-based controls, steroids, hormones, low PD molecules, environment controls. We have a lot of things coming up on HVAC, water, and other utility aspects. Transportation, cleaning validation, verification. I think we have excellent guideline already available over there. So overall focus is to make sure that the learning which industry is having, cross-functional, we bring it on the table and make sure that we provide the best document. We are fortunate to have Dr. Ranjana as a mentor. We have Dr. Sanjay Kapadia, Amit, Vijay, Sunil, Abhay as an active members of this team coming across all the reputed companies. We look forward to have our draft meeting, first cut draft meeting in November 24 with Dr. Ranjana, followed by submission of this draft in December. Very aggressive timeline, but we look forward to add that value to the industry. Thank you.
Great.
Rajiv, I just want to say something. I mean, hats off to IPA when we started with coming up with guidances, and each time we're coming up with different topics and topics that really matter to the industry. And I think this is really good for years from now, 20 years from now, 30 years from now. Once you write it down, you've created history. So I think it's wonderful, and God bless IPA for doing this. It's very good. Very good.
Thank you so much. I think we have a couple of questions here, but I think during the course of our discussion, I did try to touch upon those questions as we spoke, and most of the questions have been answered. So I'm sure that the guidance that comes up now in the next couple of months' time, we'll have more insightful discussions. This is going to be quite useful. So thank you so much for all your inputs, Dr. Ranjana, Kelia. Thank you. All the good points that you brought in about the guidances. Vipul Bhai, with your experience, I think you came up with so many good ideas. And Praveen, thank you so much for bringing that dimension about sustainability and quality culture in your.
Oh, I'll talk about it some other time. But
That's,
thank you
that's
so
my
much.
favorite. Quality culture is my favorite topic.
Yeah,
So
yeah.
as sir, we are taking a lot of steps in terms of
Absolutely,
strengthening it.
absolutely. This is something quite a unique designation, but I'm sure that you bring a lot of your experience and insight into the entire gamut of your life. So thank you so much, all of you.
Thank you.